next presentation will be on nanobodies in medical diagnostics and as new tools for reviewing old concepts by Professor Cristina Siontoro, Laboratory of Simulation of Industrial Processes, Department of Industrial Management of Technology, University of Piraeus, Greece, and Professor Siontoro.
No, that's not mine, actually. That's not mine.
I think because we are running ahead of time a few minutes, and the projectionist goes by the time schedule.
Would you like me to step down and continue with you?
Yeah, but they're missing, yeah. Okay. I think there is some problem in locating the slides. So I think we are going to switch to the next speaker. And that will be Dr. Felix Schumacher, Senior Scientist, Lead Identification, Roche Innovation Center in Penzberg, at Roche Diagnostics in Germany. So, Dr. Schumacher.
Okay, thank you very much for the introduction, and okay, cool, an earlier time slot. It's my pleasure today to share some thoughts we have at Roche for new antibody derivatives for targeted payload delivery. And I'm pretty sure if we talk about antibody conjugates, pretty much most of us, the people in the room, have something like this, a picture like this in their head. So it's Paul Ehrlich's magic bullet to which we attach a drug. I guess payload would be a better word because you can pretty much attach anything you like, DNA, analogs, peptides, small molecules, whatever you like, either via chemical conjugation or if it's a peptide or protein via genetic fusion. And because it's a magic bullet, you give it to your patient and it of course will go only straight to the tissue you want it to go and deliver the payload. Beautiful, simple idea, and probably you know all the saying, the most simplest idea are the best ones. There's just a little flaw, it doesn't really work that simple. If you look at the therapeutic landscape, so what actually of antibody conjugates have made it into the clinic, have made it into drugs, based on research of the last decades, it's actually not a large number, it's just 5 molecules. There are 3 antibody drug conjugates out there, one which has been withdrawn by Pfizer voluntarily, Mylotarg, for the lack of efficacy, and 2 radioimmunoconjugates, one which has been already phased out because it's not used anymore for therapy. So despite, I guess, a lot of research and a lot of time that it's gone into this research, there's not a lot of molecules that actually have made it on the clinic. Why is that? Well, we're doing science, so there are a lot of theories around there. One theory I think most people believe in, and we'll come back to this, centers on this number, 0.01%. And this is, if you believe these studies, the percent of injected dose per gram that actually make it to the target tissue, organ, or tumor. So that basically means 99.99% of the drug that you give to your patient is not going where it should be. And this, of course, is a problem because you can't simply increase the dose until something happens, because usually toxicity happens first before you get a therapeutic success. So how can this problem be solved? And there's actually a large number of academic groups, and pretty much every big pharma company has some people working on this problem as well. And interestingly, people focus mainly on the potency of the drug, so they want to increase potency of the drug, increase the number of drugs per payload, or per linker or per carrier, or they are switching mainly to site-specific chemistry away from random chemistry to make sure the molecules they're producing are actually those they want. While these approaches surely start to show a positive effect on the use of antibody conjugates, none of these is actually actively going after increasing the number of successful delivery events, right? So this certainly will not increase the number of molecules that actually make it to your target tissue. They simply increase the efficacy of the single delivery events. So you don't have to be an expert to see what's missing in this picture, the antibody carrier. It's amazing how little work is actually done, or seems to be done. Not everything, of course, appears on conferences and in papers. And on the carrier itself, on the protein side. And this is actually even more stunning if you think a little about design space. So if we look at the chemistry side of things, the payload, the linker, and the conjugation chemistry, your design space is usually a few dozens or a few hundreds of atoms. And most of them you can't change because they are responsible for the potency of the drug, for cleavable linker, for your conjugation chemistry. On the other hand, you have thousands of atoms in an antibody, and while I guess everyone agrees that you should not mess with the CDR regions and some structural features, there's still plenty of space to play around on the carrier side of things. Okay, what would that be? I believe there are four main aspects one needs or should be looking at if we think about engineering antibodies as payload carriers. The first one is the so-called binding site barrier. I will come to this in a second. The second one is, of course, very closely connected. This is the idea of tumor penetration. How deep can you get with your molecule? And as I said, these things are very close connected, but tumor penetration there has a little more aspects than the other thing. The third one, and probably most important one, is PK. I guess this is one of the most or major advantages of antibodies. They do come with a good PK, and from this good PK you can start to make it better or worse. And this is something many other approaches like aptamers or alternative scaffolds don't have, and often people eventually have to start with these approaches to engineer PK in there. Antibodies start with a good PK, so you should not lose it by attaching certain payloads. And then the final thing I've called here biophysical properties. This for me comes really down to candidate selection, which should not focusing alone on affinity, something we have been doing over the last years. There's so much more physical or important properties of antibodies and carriers that play a role if a certain molecule is a good delivery vehicle or it's not. And you should take this into account. I could, of course, talk for quite a while on all of these aspects, but you would be bored to death very soon. So I've decided to simply take one of these as an example, how these ideas could be turned into reality in terms of engineering. So for those who probably don't know, the binding site barrier is a theory that tells us that the antibody, if it has a decently high affinity, will never make it actually across the perivascular space or beyond the perivascular space, because as soon as it sees its target, it will bind it, it will never let loose again, and therefore it will be internalized and degraded. So you basically can't get into your tumor very efficiently. Well, if this is a problem of affinity, it seems to be a relatively easy to solve problem. Simply go down with the affinity and get better penetration. Problem is, of course, at the same time you will lose selectivity and most likely also internalization, at least when your K-off rate becomes faster than your internalization rate. But you can overcome these problems, of course, by increasing the avidity. So if we go down with the affinity and increase the avidity, we should get good penetration, solve the problem of selectivity, and even also internalization. But since we are already engineering, we can even go a further step, and Professor Jane has already mentioned it in his introduction. We can do these two things and then add specificity. So add specificity for a different target, go for bispecifics, for example. And there's a beautiful work done by MedImmune in this space, not so much, unfortunately, published yet, but I'm really looking forward to that. And a molecule with this kind of properties should actually have really good penetration, high selectivity, and recent literature actually suggests that if you have more than one specificity, you should actually also have an improved internalization rate. So if you put that into an engineering approach, how could that look like with antibodies? So we start with a magic bullet, IgG, go down with the affinity, add a specificity. At Roche, we have this beautiful bispecific format we call cross-map. It looks like an IgG, but it basically has two different Fabs targeting, of course, two different targets or two different epitopes on the same target. And to add increase the avidity, we simply can add another binder at the C termini. And if we can add one, why not add two? So we have this 2+1 and 2+2 formats. And both of these molecules actually would suffice these engineering principles we've just come across. And then, of course, this is very simple. You can then start to introduce the cross-map technology into this 2+2 format and go completely crazy with specificities, affinities, and that sort of stuff. Now, crazy is very important because, again, this is a nice idea which looks nice on paper or an electronic screen, but I guess everyone here is aware that this is not a simple task to do. So it's very, very tricky and difficult to dial in all the right affinities and targets and that sort of stuff. But it can be done. And I think if we go back to these four aspects of antibody carrier design, these molecules will certainly make a better carrier than IgGs. But, of course, there's another problem. These design aspects, of course, are also closely related. So if we go with a molecule like this, you immediately see the size has increased, and that is probably, again, negative for tumor penetration. To solve this problem, we are fortunate at Roche because our leadership decided to acquire a company called Dutalis. And these guys have developed a very interesting technology where they managed to make bispecific Fabs. This works that way that these guys managed to make Fabs where 3 of the CDR loops are actually efficient or sufficient to bind a target. So you have 3 left, which you then can optimize to bind the different targets. So with a bispecific Fab in hand, if you then go back to an IgG-like format, you now have a molecule that looks like an IgG, but it has 4 binding sites, 2 specificities, and you can, of course, dial in the right affinities. And now this, of course, looks like a good starting point, like a molecule that might be poised to overcome the binding site barrier. And now you can start to optimize it for these other aspects. This actually brings me to my last slide. I hope I have shown you, or even convinced you, that why we believe that antibodies are still good vehicles for payload delivery. And if you imagine for a moment that this box is all of the design space where we can get with antibodies as delivery vehicles, with all the engineering technologies we have, I believe that the starting point we had with a standard IgG was probably not a good starting point for a delivery vehicle, and that if we increase the design space only on the chemical or payload side of things, we might not increase this design space large enough to get an overlap with the area where we see actually therapeutic success. But by engineering the carrier, we simply move this starting point to a completely different place in the design space, and from there we hope to find optimization pathways into an area of therapeutic success. And what I mean here is not that we are succeeding in getting 1 or 2 molecules on the target, on the market, like what happened with the ADCs. I mean, Kadcyla, etc. These are beautiful molecules, but the ADC pipeline is not looking very good at the moment. But what I mean with this is really that we find a number of molecules that share certain properties in engineering, and then they are different in other properties like payloads and targets to use them for different indications. And this is actually what we think about antibody engineering, and hope that we then, with these ideas, come closer to the slogan the company has given itself. Thank you very much for your patience, and I'm happy to take questions.
So any questions? Okay.
Do you have any data that shows that increasing the avidity actually helps overcome the binding site barrier? I would think it would be just the opposite.
Yeah, the data is still missing. There is this interesting concept you may have heard of avidity-driven selectivity gain, and on back of that, you can see effects like that. Yeah.
Penetrate better.
No, that's still a theory.
First, I would like to thank you. I think it's very valuable that Roche, companies like Roche also tell us what's ongoing so in the early phases. So I think that is very useful for the whole community. Then a question, I'm sure that when you are working in such a company, you are thinking about which challenges will you have to make reproducible batches of such compounds for getting it to the market. Can you comment on that, both regarding at the stage you are now, and if you would like to add drugs to the compound?
Yeah. A perfect question. Thank you very much for that. I think, so I guess there are a few aspects to this. One point is I'm coming from the biology side, so I'm in the large molecule research, there's my home. And it's actually amazing how far we have already driven the technology to make these cross maps and 2+2 formats in a robust and repeatable way and have the CMC really on a GMP scale level. The thing is, as you probably know, Roche itself has not a massively large experience with then coupling things onto it. The most experience is on the other side of the Atlantic at Genentech. But of course, we are one company and we're talking to each other. And this might sound like a bold claim, but I'm pretty sure, as it is at the moment, that we should be able to really get these things right, especially with all the improved technology on site-specific conjugation that is coming out now, where if you look closely, the really good ones are actually based on protein engineering as well, like putting tags for conjugation at the right position, and then simply sticking on the chemistry side of things. But it's a very important aspect, yes, definitely.
Thank you for the very nice presentation and your approach. I looked at the list of currently available and development ADC, but none of them are very small. So the question is size-wise. You have an antibody already and you combine the drug. How much of that is somewhat a disadvantage overall, and what sort of approach you might be doing? Of course, we haven't heard the presentation on antibodies yet, but in this approach, how can, because that's one factor in delivery.
Yeah.
Although apart from targeting, but also size, and it has something to do with the penetration as well.
Yeah, very good aspect. To keep the answer short and keep in time, size and penetration are always connected to PK. So if you have a very small molecule that penetrates very well but has a very short PK, that doesn't help you a lot. There is a beautiful study out there, I can't remember the author right now, but they show a single chain VF, Fab2 primes and full IgG, how much actually is delivered. And the fastest delivery is definitely by the single chain VF, but the overall delivery, the most delivery is done by the whole IgG. So if you have a molecule that is small, but has a PK like an IgG, that would be ideal. And I'm unfortunately unable to show this, but we can decrease the size of the molecules with keeping most of the good PK properties of a full IgG.
Good, thank you.
Okay. Thank you.
Did we find the slides yet?